Imagine a world where we can custom design biological materials, revolutionizing fields like medicine, agriculture, and biotechnology. This isn't science fiction, it's the reality of recombinant DNA technology. So, what exactly is recombinant DNA technology? Well, it's a fascinating process that involves combining DNA from different sources to create entirely new genetic sequences. This groundbreaking technique opens up possibilities that were once thought to be impossible. Let's delve deeper into some key concepts of recombinant DNA technology. First up, DNA cloning. This process involves creating identical copies of a specific gene. How do we do this? By using restriction enzymes to cut the desired gene, which is then inserted into a plasmid. That's a small circular DNA molecule. These plasmids act as vectors, carrying the target gene into host cells, such as the commonly used E. coli bacteria. Once inside, the host cells replicate the gene, potentially producing valuable proteins like insulin. Speaking of restriction enzymes, think of them as molecular scissors that recognize specific DNA sequences and make precise cuts. After the gene is inserted, DNA ligacy comes into play, acting like molecular glue, sealing the nicks in the DNA to complete the insertion process. Now, let's walk through the steps of recombinant DNA technology. First, isolating the gene. This crucial step involves using restriction enzymes to cut the target gene from the source DNA. Next, inserting the gene. Here, the cut gene is carefully inserted into the plasmid vector. Then comes transformation, where the recombinant DNA, that's the plasmid with the target gene, is transferred into host cells. This transformation process is pivotal for the next step, which is product extraction. In this final step, the product, such as a protein synthesized by the host cell using the target gene, is extracted. This protein can then be used for various applications, depending on the gene that was initially targeted. Now, let's delve into some revolutionary applications of recombinant DNA technology. Starting with biomedical applications, this technology has paved the way for advancements in gene therapy, enabling doctors to correct genetic disorders by inserting new genes into patients' genomes. It also plays a crucial role in the diagnosis and prediction of diseases, providing tools that help identify genetic disorders before they manifest. Next, let's talk about insulin production. Through recombinant DNA technology, insulin, which is essential for treating diabetes, can now be produced in large quantities, making treatment both more accessible and affordable. Lastly, we touch on transgenic organisms. By integrating DNA from different species, scientists have created transgenic animals and plants, which contribute to research, agriculture, and even pharmaceuticals. To wrap up, the importance of recombinant DNA technology cannot be overstated. It has revolutionized the fields of medicine, agriculture, and beyond. I encourage you to explore this fascinating field further, as it holds the potential to significantly impact our understanding of biology and improve countless lives.